All right, everyone, let's get going. This is sadly, the last session already of the conference. I hope it's going to be a great last session. My name is Felix Bayer. I'm a postdoc at the Max Planck Institute for Brain Research in Frankfurt. I'll be chairing this session. The last session is dedicated to ecology and evolution, so we will be hearing from 4 amazing speakers on how changing environments to what extent changing environments can drive evolutionary processes and how they may affect the ecological interactions between individuals and species. This work is going to span a diversity of techniques. We will start in birds. We will then also hear work on sticklebacks, on social insects, and we will finish again in fish. Sorry, Hans. Yeah, exactly. That's the one thing you don't want to. But our first speaker is Carlos Botero. He is an associate professor of integrative biology at UT Austin, where he uses theoretical models, large-scale comparative analyses, and experiments in the field in the lab to link changes in the evolutionary process to selection fluctuation. Welcome.
Thank you. Thank you, Felix, and thank you for hanging in there. Thank you to the organizers as well for this incredible opportunity for exchanging ideas and hopefully start new collaborations. I am somebody with slightly different backgrounds to the speakers that have come here before me. I'm an evolutionary ecologist, and for the course of my career, I've been really interested in understanding how the repeated exposure to, or how animals cope to the repeated exposure to environmental variability and unpredictability. And because in many ways animals do that by changing their behavior, over the years I've become increasingly interested in understanding how changing environments have shaped the evolution of the brain and how changes in the brain, conversely, have shaped organismal abilities to respond to novel environmental challenges. Now, what I want to do today is to focus on the second question today, probably, as a way to give you a little insight into the kinds of tools and techniques we use, the kinds of questions that we ask, but mainly to showcase the main challenge that we found so far and what we hope is an exciting way to address it that we're starting to do right away. And I want to start by talking about this foundational concept in cognitive ecology known as the cognitive buffer hypothesis, which states that probably one of the main reasons why bigger brains and enhanced cognition evolved was because animals were exposed to more variable and more unpredictable conditions. Now, the idea behind this is that when conditions are difficult to predict and when they're changing all the time, it is very useful to have some behavioral flexibility. And we don't have to go too far. We can see it in our own species. The product of accumulated knowledge, problem-solving abilities, learning, innovation, all those kinds of traits allow organisms like ourselves to occupy and explore environments to which our phenotype is clearly not optimally adapted to. But we can generate or we can extrapolate this idea to all of the animal world by thinking about, for example, a given physical trait that needs to be matched to an environmental target. For example, imagine the amount of insulation around one's body such that lots of insulation is really beneficial for cold places and really costly in warm places, and little insulation does the opposite. Now, if you take a population that has been adapted to a specific environmental condition and we expose it to an environmental change, what you find is that this gradient in fitness drives immediate directional selection, taking the population or removing from the population individuals that are more mismatched than others, and drives a quick evolution towards the new optimum, and the population kind of like oscillates thereafter. Now, if we introduce to this a cognitive buffer, for example, something that could happen by allowing the possibility of choosing more strategically microhabitats or being able to learn or have like flexible foraging techniques that allow you to encounter and produce enough energy to overcome all the extra challenges of thermoregulation because of your new mismatch, then you find that the gradients become a little bit more muted and the speed at which the population adapts becomes a little bit slower. However, after a short time, the populations basically are completely undistinguishable from each other, and that's something important to keep in mind. Now, you can theoretically ratchet that buffering all the way to 100%. And in that case, there is no longer selection. The phenotypic value is completely inconsequential, and populations no longer show an evolution of the mean, but they do show that over time, both within and among populations, there's a radical increase in the variability that we see. So having started from this thought experiment, then we can now start asking the question, well, what do we expect? What are the consequences of developing better abilities, better cognitive abilities? The first thing we already saw it, and it's that initial rate of evolution slows down roughly in a linear fashion. The second thing is that the amount of genetic variance that you see in the population is going to increase. The standing genetic variation increased dramatically so, particularly towards the end of the spectrum in cognitive buffering. And when you think about it, the availability of more genetic variants coupled with the idea of individuals that are able to withstand environmental change more easily or that pay less costs about that, leads us to the third consequence, which is that populations become much better able to deal with acute environmental stress. And by the way, the threshold at which we go from extirpation or extinction all the way to survival depends on the extent of the challenge that these populations go in. So the stronger the challenge, the farther up that threshold becomes. Now, the thing that I really want to point out here is that if you think about it, we're seeing kind of like two very opposite forces. One is that we're saying that selection is, or that the rate of evolution is slowing down with the ability to buffer the environmental selection. But the other one is that the opportunity for evolution to happen is actually increasing with cognitive buffers because these populations are better able to go to places that others are not, or to survive changes that they're not really, to which their phenotypes are not really suited for. And that drives us to a very straightforward prediction, which is that the highest potential for evolution should actually be occurring at intermediate levels of cognitive buffering, because at that level, buffers are strong enough for populations to be able to explore more environments, to withstand more change, but they're not too strong to prevent evolution from happening altogether once they reach those environments. Now, theory, of course, is only as good as the predictions that it makes. So in my lab, we've been using studies, comparative analysis of brain size to try to understand what, if any, of these components are actually true. And fortunately, I wouldn't be standing here if it turns out that it was not true, but fortunately, most of the components seem to be very well supported. For example, we find that was not what I wanted. All right, for example, we find that indeed species or populations that have bigger brain sizes tend to evolve much more slowly than populations with smaller brains. And this we found by following the changes in body size in passerines over the last 50 years, through which global temperatures have increased and body sizes have tended to be reduced, basically an expression of Bergman's rule, because smaller body sizes facilitate or increase surface to volume ratio and facilitate heat transmission. But we found that the speed at which evolution happens is much more slowly, a threefold differences in big brain birds than in their small brain counterparts. Similarly, we found that bigger brains have actually allowed populations or species that have them to be more resilient to environmental change. So they exhibit less mortality during environmental extremes and more stable populations across the gamut of environmental conditions that you see. And most interesting, we've actually found that when you look at the evolutionary transitions from bigger to small brains and from variable to stable environments and the other way around, it turns out that there is pretty good evidence, at least in birds, that it was the evolution of big brains that facilitate the colonization of variable environments and not the other way around. So against basically this idea of the cognitive buffer hypothesis we were mentioning initially. Now, I could go on and on with other kinds of studies that do this, but we don't have time today to go into any one in detail, so I'm going to stop with the examples here. But I do want to point out that one of the key things that we've learned, or that there's many really interesting phenomena that we've been able to pick up with through these methods. But one of the most frustrating things with this particular kind of work is that after all, brain size is really a huge, big black box. And then at the end of the whole day, we end up realizing that yes, we have some ideas about what things may be happening, but we don't really know what are the conditions what are the cognitive abilities that are being lost or being gained as these processes unfold. A much more informative process, ideally, would be to actually like open that black box and illuminate completely by trying to figure out what are the underlying neural networks inside those brains to try to kind of like get a better sense of, first, how do neural network structure relates to function, and how has the architecture of these networks changed over time. That would be a good target of research goals to do that. And fortunately, in recent times, we've actually had advances in fMRI tools and other technologies to try to do whole brain imaging that allows us to do functional connectomes at the species level. The problem with fMRI, however, is that it's really expensive. It's virtually impossible to take it to the field, and it requires that animals stay really still during imaging, which means that you have to spend, for this little pigeon, this guy actually had to undergo 2 months of training, slowly going into, until you get to the point in which they can tolerate the noisy and cramped conditions of an fMRI, and sometimes they actually fall asleep when they do that. So the alternative is to just kind of like put these animals to sleep with anesthesia, but then that disrupts all the patterns of functional connectivity that we're really after. In response, I've kind of like assembled a team of rock stars, except me. And these rock stars are actually radiologists, avian neuroscientists, and AI computational scientists. And what we're trying to do is to provide an imaging alternative to produce this whole brain functional connectivity data that we would like to collect in hundreds, hopefully thousands of species in the next 10 years. The principle of what we're trying to do is that we're replacing MR with light. And if you have a strong enough light source, it can penetrate bones and tissue, and we can use it to image oxygenated blood in very much the same way that we do that with magnetic resonance right now. This technique, by the way, has been developed really well for, this is actually an old picture, things are much better, and they're wireless right now with humans, but this has been developed in humans, and a similar but less involved technique has been used in mice and rats. Now, our project, which is currently funded by the Human Frontier Science Project, program involves 3 different stages. We've completed the proof of concept, and we're now creating a true optical tomography device. And what I want to do today is kind of like to tell you a little bit about kind of like how that project, or that first proof of concept, what we did, and what we're doing right now. Now, as a proof of concept, we're using a technique known as optical intrinsic signaling, which involves a single light source and a high-resolution camera. It allows you to image a specific region of the brain, and it allows you to only image in 2D. That's a big limitation. But the nice thing is that all the steps post-image acquisition are basically the same of what we're going to do in a 3D setup. It involves basically some signal processing, some denoising, some analysis to eventually produce these parcels, and then the parcels, we use them to create the networks that we're after, or to characterize the networks that we're after. And so far, we've done that with about 40 different pigeons, and we've gotten really consistent and very exciting results. For example, when we do these imaging sessions on a single individual, we find the same results on, say, like the first 20 minutes of recording than the second 20 minutes of recording. By the way, these are our correlation matrix of the activity, where red means, or darker colors mean stronger correlations. So we basically have consistent characterizations of these networks in multiple different imaging sessions of the same individual. Additionally, perhaps more importantly, we find that pixels in our field of view that belong to regions that are anatomically defined, that we know that work for specific purposes, say, for example, the visual walls or the hippocampus, those appear to be much more strongly correlated, which is what you would expect. And most importantly, when you organize these matrix by hemisphere, you see that there's strong homotopic correlation. So the regions in one side of the brain really correlate in activity with the regions in the other side of the brain. So all of this is quite encouraging, that we can do this just using light. But ultimately, what we want to make sure is that we can produce something that is equivalent to the gold standard, which is this fMRI technology. And the nice thing is that we do. So this is an aggregate of 6 individuals that we image using fMRI. And the regions that we're providing here and the parcellation that we obtain, were created by looking only at the surface data to make it comparable to what we're seeing with OIS. And what you find is that across all the different individuals that we've imaged in slightly different ways, you find very consistent results, very similar organizational structure that we characterize from this data. And more importantly, when you look at the patterns of connectivity that we derive from OIS, which are here in the left, and we compare them to the patterns we derive from that average fMRI data set, we find basically the same things. And in fact, we find that the correlations are much stronger because of that high resolution camera with OIS. Now, the next step, or the next thing that we want to do, by the way, is to just take this data, or take this technique, and turn it into 3D. So the first step, for example, involves going from one source and one receiver to 16 sources and 21 receivers to create a true optical tomography device. We are in the first stages of this process. We have already started with a smaller array, and as you can see here, this poor little pigeon with nice warm blankets being imaged. But the nice thing here is that at the end of the day, you just remove that little cap, and the pigeon can just walk away with no problem. So this is a non-invasive, portable technology, easy to replicate, and that produces imaging at a fraction of the cost of what you would do with fMRI. And even though I don't have right now actual data to show you for the 3D version of this project, what I can show you is that the simulations, light spread simulations, indicate that we will be able, so red is kind of like better ability to discern processes, indicate that we will be able to image the entirety of the avian brain using this setup with 16 sources and 21 receivers. Eventually, we want to go up a little bit more, but like at the very least, 16 to 21 seem to be working really well. All right, so the ultimate goal of this project is to actually characterize, the dream of this project is to characterize the functional connectivity network in hundreds of different species, hopefully thousands over the next 10 years. And this might seem like a pie dream, and it maybe is. So that's why I'm making a call for all of you guys that are really interested in exploring potential issues about functional connectivity, parcellation, interspecies comparisons. Please reach out to me and please become part of our team. We want to make this a community, an open access to the community. We want to make this a community effort. We believe there's incredible opportunities to advance even basic sciences, like asking questions like how does neural structure relate to neural function, asking questions about, well, how do functional connectivity assays really predict what we see, how repeatable are these networks over time within individuals and among individuals, how much variability, millions of questions to ask. There's also other things that we would like to ask, which are like, well, what is the best way to compare across species? There's a lot of really interesting theoretical and practical challenges that we will be dealing with, and I would like to open it to the community to do just that. So if you're interested in what is going to be our stage 4, which is going to happen probably within 2 years, please reach out to me and please let's start working together. And I think I have plenty of time for questions.
I wanted to remind especially our early career researchers that this is the last session of the symposium, so please, your last chance to ask questions. Do we have any questions?
Great work. We started to use functional ultrasound imaging, high resolution, for the same purposes, to look at the whole brain, neurovascular coupling. And one of the problems that we currently experience is the reference, brain atlas reference. So how do you overcome this problem for your imaging technique? Do you have to create actually your own brain atlas all the time?
So for the proof of concept study that we did, we chose the pigeon because we have an actual really good map or good atlas for the pigeon, and we also have an actual anatomical connectivity map that was created by one of my collaborators. So we use kind of like the golden standard that we had. But part of the things that we're trying to figure out in the future is how to do that on species for which we know nothing about, which is what's going to happen very soon. And one of the things that we're discovering is that basically some of the choices that we do during the analysis are very consequential. So the AI scientist, this South Korean guy, Yoon, we're starting to work on those kinds of questions. How do you actually parcellate the brain without expert knowledge? And we hope that we're going to be able to use the data that we have with the pigeons to try to train some models to assess that in the future. Yes, please.
Any other questions? Yep.
Very cool stuff. Maybe you mentioned this, but what are you actually measuring with the light? Like, what's the signal that you're picking up? And I was wondering, because is this something that's specific to vertebrates, or could this be applied to, say, butterflies that you have millions of samples that you could access or so?
So the principle is basically the same as fMRI. Every time that a neuron fires, there's a neurovascular kind of like pulse of oxygenated blood. And so that coupling is what allows us to, by just paying attention to the color in the image, we can tell when oxygenated blood is pumping through the system, and that basically indicates that there was activity in that part of the brain. And because neurons that tend to be connected to each other are more likely to spontaneously fire at the same time, we can estimate the strength of the correlations between different neurons by looking at the correlations between the patterns of oxygenated blood in different parts of the field of view. So this probably is a vertebrate-oriented system, unfortunately, but...
Is this similar to the intrinsic optical signal in a way? Because with that one, you would pick up density differences, and that one, I think, applies to all brains, not just because it doesn't require blood movement.
Yeah, so it is part. So it's part of kind of like all related techniques. Yes. So the optical tomography basically is based on very similar principles. All we're looking at is oxygenated blood or changes in color. But if there's something that drives changes in color, we should be able to detect it through these techniques.
Okay.
I'm curious, for aquatic species that need to remain submerged, imagine you could immobilize them. Are there issues with optical properties in water that would preclude the use of this technology? Or how do you envision the potential of this technology for working for aquatic species?
Well, so light is traveling through some liquid in the brain and like humans and everything. So the key thing is, a key aspect of this technology is that you need really good coupling between the light sources and the thing that you're measuring. So with humans, we kind of like have this like special springs that make sure that all the sources really are close to your thing. So we can go through hair, through skull, through those kinds of things. With birds, we're kind of like working on how to go through feathers, all this kind of stuff. But it really matters that you have like good coupling to avoid diffusion of light that would create confusion for the system. I imagine that if you could have good coupling with a fish, it doesn't really matter whether you're in an environment that is aquatic around, because there's no environment in between the source and what you're measuring.
Yeah, so you just need a good fish hat.
Exactly. Yeah, you would need a really good coupling between the cap and the animal.
Okay.
And by the way, so the other thing is that it's a key thing is that there's limitations in terms of size. So if you want to do tomography, it's harder to do with really small animals, really very small animals, because you don't have enough channels like enough sources that you can fit into that. We are working on some miniaturization components to this work. We think that for a wide variety of the birds that we can work with, that's not going to be a problem, but it could become a problem the smaller you go. So I don't envision this, for example, on a zebrafish or something like that. We just can't do that.
Okay, we have one more question from Sean.
Yeah, so this is something I've been talking to other folks about as a kind of a general issue, and I hope we have time to explore it more. What do you view as the potential for making this mobile and transportable to remote field locations? Because, a lot of the birds, say we're going to stick with birds, are not going to be able to be brought to Texas or whatever.
Yeah, so that is a critical component of the design of this system. We want it to be cheap. We want it to be repeatable, non-invasive. We want it to not involve any anesthesia, obviously, so that we can measure this connectivity without disrupting it. And we want it to be mobile. the mobility of it is, so right now it's kind of like, I could give you the pitch and say like, yes, this is fully mobile, and we hope that it will be, but it's still, when I'm saying mobile, I'm envisioning more right now, kind of like taking it in a van to the field, capturing the guy, bringing it to the van, and doing it right there. I don't envision somebody like carrying a little thing and then just doing it, but it's sufficiently, like way more mobile than an fMRI. Like we can take this to the field, and my goal, for example, is to go, so as soon as we have this, kind of like my there's this zoo in Spain that has 89% of all parrot species in one location, so like 380 different species. I'm hoping to take this to that zoo and then go to town every day, measure a different species, and within a few years have an entire order to do these kinds of evolutionary analysis, but at a very mechanistic level. That's kind of like my next goal, wherever the wood is.
Thank you very much, Carlos. That was
Thank
great.
you very much.